Okay, so our next speaker is continuing our little schedule shuffle. This is Dr. Shyamalika Gopalan, who's joining us from Duke University in North Carolina, and will be telling us all about hunter-gatherer demographic trajectories. So I'm really excited to hear about this. Take it away whenever you are ready.
Is it on?
It's low battery. So
oh no, sorry, the mic.
I think, did you put the button on the side?
Okay, is that better?
That's great.
Okay, awesome. Yeah, hello everyone. I'm very excited to be here. I've been really enjoying all the talk so far. And yeah, thanks for having me at your session on small populations. As somebody that studies mostly human genetics, not a rare species at all. But actually my work today, the project I'm going to speak about is actually about small human populations. So I'm going to be discussing my work on hunter-gatherer demographic history during the transition to agriculture, specifically in Eastern Africa. And this is work that I did as a PhD student in Brenna Hen's lab. So the focus of my work that I'm going to be speaking about today is actually this part of the world, Southwest Ethiopia. And so here, this part of the world currently is home to some of the only hunter-gatherer populations left on the planet. And the transition to agriculture in this part of the world is actually still ongoing and began relatively late compared to in other parts of the world. So as we all probably know, at the beginning, all human societies subsisted exclusively on hunting and gathering. And then at some point, starting about 12,000 years ago, in fact, groups of humans began to transition to relying mostly on domesticated plants and animals as their primary means of subsistence. So I'm really fascinated by this process historically, the mechanisms by which agriculture spread throughout the world. And today, of course, most human societies are in fact based on agriculture. And so for this reason as well, I think it's really an interesting opportunity to be able to study this part of the world, Southwest Ethiopia, where there are actual living hunter-gatherers left. And we can sort of study this living population in a much more deep context than, for example, with ancient DNA. So again, the focus of this work is this population of hunter-gatherers, the Chabu. And so they live in these very densely forested highlands of Southwest Ethiopia. And here they practice many of their traditional sort of life ways, including hunting wild game and gathering honey. But at the same time, more recently, in the last few decades, they have become more settled into permanent towns. They've begun to take up more small-scale farming and just generally become more integrated into the regional economy there. And unfortunately, a lot of these changes coincide with the displacement of the Chabu people from their traditional lands, primarily actually by coffee plantations. So you might be able to tell that this is really prime real estate for coffee, for growing coffee. And so that has been a major pressure on this population. So through collaborations with anthropologists, we were able to sample 83 Chabu from Southwest Ethiopia. We were also able to sample some neighboring populations that live nearby, specifically the Majang, the Bench, the Sheko, and the Shikacho. And so here I've also noted that the Majang speak a Nilo-Saharan language, that's what the NS stands for here, whereas the Bench, Sheko, and Shikacho speak an Afro-Asiatic language. And so these are the two major language families that are prevalent, and this detail is somewhat important for some of the results later on. Interestingly, the Chabu actually speak what is thought to be an isolate, so that's sort of another interesting data point there. And then additionally, we have a sample that was published in 2015. It's actually the first African ancient genome that was ever sequenced and published. And this is this individual that's known as Bayira, happens to be found quite nearby a region of interest. And importantly, this individual lived about 4,500 years ago, which is well before any evidence of farming, of pastoralism in this region. So we can be pretty confident that this individual was indeed a hunter-gatherer. And in addition to those data, I also integrated published data from across northeastern Africa, as well as from West Africa and the Near East, just for more complete genomic context. And so, one of the first analyses that I did with this data set is to just look at patterns of population structure and look at factors that might be structuring these groups of populations. And one of the first things that jump out here is that in this context, genetic ancestry is actually very closely correlated with linguistic affiliations. Not perfectly, there are some exceptions here, but generally speaking, populations speaking Nilo-Saharan languages tend to cluster together, whereas populations speaking Afroasiatic languages, again, tend to cluster together. And particularly, we identified sort of this yellow component associated with Nilo-Saharan speakers being at high frequency in Nilo-Saharan speakers, and this green component in the Afroasiatic speakers. And so I didn't really explain this, but we've seen a few plots like this so far. But this is the program admixture, and typically in human studies we use tens to hundreds of thousands of genetic markers, independent genetic markers, to be able to sort of parse this structure. And so looking at sort of these Southwest Ethiopian populations again in this area of interest, what we see is that there is a really high prevalence of this additional genetic component here in sort of depicted in this purple color. And this genetic component is at very high frequency in this ancient individual that I mentioned, Bayera, who again lived prior to any evidence for farming in that region. And in addition to that, we see that the Chabu are also sharing this genetic component at a very high frequency in this population. And it's really prevalent throughout this region. And so, we can say that the Chabu are very closely related to this ancient hunter-gatherer individual. And this is something that we also confirmed with principal components analysis. So here you can see that the Chabu are sort of defining the corner of this triangle in PC space, and that Bayera, in fact, lies very close to them. And so looking across, this entire region, we can say that these populations in Southwest Ethiopia are really the result of gene flow among multiple different groups. So we have this component associated with ancient hunter-gatherers, as well as these Afroasiatic speakers, this green, and Nilo-Saharan speakers in yellow. And I also confirmed this hypothesis of gene flow using more specific tests that specifically test for gene flow. So not simply looking at admixture patterns, but also F3 admixture statistics, for example. So when we plot these genetic components spatially across the region of northeastern Africa, we can see that, for example, the Afroasiatic component on the far left here and the Nilo-Saharan component, they do overlap in space, but they do sort of have these fairly defined regions. And these actually correspond quite nicely with sort of archaeological evidence that we have for the transmission or the spread of crop agriculture and pastoralism, respectively. So the movement of wheat and other crops associated with Afroasiatic speakers, and the movement of pastoralism with speakers of Nilo-Saharan languages. And then additionally, again, we see this sort of hunter-gatherer or Bayera associated ancestry kind of wedged in between them, and that's sort of where the Chabu are and where Southwest Ethiopia, these other populations are as well. And what's interesting is that with the Chabu, we actually see relatively little evidence of sort of historical gene flow with either Afroasiatic or Nilo-Saharan populations. Although more recently, anecdotally through the ethnographic evidence, we know that they are intermarrying with other groups more frequently now. It seems that in the recent past, they were more isolated. And so the next thing that I was interested in looking at were sort of to look at these sort of signatures of isolation that we've been talking about a little bit throughout this conference so far. And so I looked at runs of homozygosity in the Southwest Ethiopian populations. And so what I found is that indeed the Chabu have the highest levels of homozygosity going genome-wide. that is compared to all of these other populations here. And that, of course, again, it indicates a small effective population size. But what was really interesting is that when I parsed these runs of homozygosity into these long and short segments, actually using a method, Garlic, developed by Zach Speech, we see the same sort of pattern recapitulated with the long segment, so the Chabu being the highest, or close to the highest of this group. But not with the short segments. So with the short segments, the Chabu are actually not particularly remarkable. In fact, we have this Afroasiatic speaking agriculturalist population that has sort of the highest levels of ROH in these short segments. So this actually indicates that this feature of having a small effective population size is due more to recent demographic events rather than sort of a long-term small effective population. So in order to be able to look more with more temporal precision, I turned to a method called IBD-NE, which looks at identity by descent, which is genomic sharing among pairs of individuals rather than within individuals, within haplotypes within individuals. And so with this method, we're promised to be able to look at the effective population size trajectories over time. And again, in a time frame that is particularly relevant for the spread of agriculture in this area. So when I applied this method to our data, I found that the Chabu population has been recently declining in size, of course. So here along the x-axis, we have time going from the past to the present, going from right to left. And you can see that the decline in population size really appears to have begun about 30 to 40 generations ago. And prior to that, we see actually that the population was relatively stable. So 30 to 40 generations in human time, that corresponds to about 1,000 years ago, which again sort of fits in with the timeline that we're thinking about. Additionally, the Mahjong, so the neighboring population of the Chabu, have also been declining in population size over the same sort of general time frame. But what was interesting is that the Gamus have not been declining. They've been holding steady or potentially even increasing in effective population size. So these three populations are actually, from our genetic analyses, are quite closely related. In fact, the Mahjong and the Gamus are almost indistinguishable in sort of admixture, in our admixture plots, and yet they exhibit these quite divergent population size trajectories. So on the other hand, we see that these agriculturalist populations have actually been increasing in size over the same time period. So here we have the Ari cultivators, the Shikacho and the Woleta. And I'll just note that these two populations on the right are plotted on a log scale. So these populations are really growing exponentially. And then finally, the last population that I'm going to show here are the Ari blacksmiths. And this population has been also declining in size in a manner that is similar to the Chabu. And again here, we have a population that's very closely related to what I have previously here, the Ari cultivators. Previous work has shown that these populations diverged very recently, and yet we see, a divergent demographic trajectory in these two populations. And that's something that we found very interesting and we're interested in to dig into some more. But for now, what I'm going to talk about a little bit are some of our simulations sort of validating these results. So I actually didn't put this up here, but I did have sort of the sample sizes that we had for all of these populations. And so for the Ari blacksmiths, this was published data, and we had only 17 individuals. So people might be concerned, and reviewers were concerned that, this may not actually work with such little data. So what we did was actually simulated some demographic histories using MS Prime. And so here I simulated a declining population size, a steady population, and an increasing population. And what I did was to apply this method, IBDNE. So if we have 50 simulated samples, we can see that we actually recover the true demographic history pretty well. So it's actually, working quite well. Like, we can be confident we have at least 50 individuals. And for most of the populations that I presented, we have about that. However, if you go down to 20 samples, which is sort of analogous to the R.E. Blacksmith's example that I showed, we can see with a steady and increasing population size, we can no longer really tell what's going on. Even sort of qualitatively, these answers are wrong, showing that the steady population is increasing and some weird bumps going on here. However, what's kind of interesting is that if we're trying to infer a decline in population size, we can actually do this robustly with as few as 20 samples. So that is actually kind of reassuring, and we looked at this across several simulations, and it seems to be wider confidence intervals, but generally we get the right answer. Okay, but one other sort of caveat to this method in general, and especially with considering small populations, is that gene flow can kind of throw a wrench into all of this. And so another, as I mentioned, many of these populations have exhibited or have experienced gene flow with diverged populations. And so I simulated a pulse of gene flow at 10 generations ago, 30 and 50 generations ago from a diverged population. And here I think it was 25% what I'm about to show. And again, it's a steady population size, so nothing actually going on in terms of population growth or decline. But with gene flow, we sort of see a whole set of different kinds of errors with IBDNE. And I think some of our previous talks, this is not overly surprising to anyone. But when we have gene flow, we have all kinds of spurious bumps in NE being introduced. They tend to be around the time of gene flow, although not always. And so here I'm showing a sample size of 20 individuals. And so, this is not very good, obviously. So we did a whole bunch of these simulations and found that really the factors that exacerbate these errors are how recent the gene flow was. So the more recent, the worse it was. And you can see that here. We also looked at different proportions of gene flow. So here I'm showing 25% gene flow from a diverged population, but if you go to 10%, it's less severe. 50%, it's more severe. And then additionally, we saw that if we increase the sample size that we have, so if we look at 50 individuals instead of 20, we can somehow, we can start to get a little bit better. And so these errors become a little bit less severe. Still, however, something to definitely keep in mind. So to wrap up this part of my talk, we found that IBDNE is actually quite a useful method for inferring past effective population size on this temporal sort of scale, even from relatively small sample sizes. Although, of course, in cases of recent gene flow, further investigation is definitely warranted, and if we think that this is going on, we should treat these estimates with a lot of caution. Overall, however, and I think what is maybe particularly relevant for this group here is that IBDNE is actually quite robust when it comes to inferring population declines in particular. So even when we have few samples, even in the case of gene flow, if we see a decline in population size, we can be reasonably confident that actually did occur. And there's sort of other pieces of evidence that we can use. So for example, the total amount of IBD is something that I didn't really get into here, but that's another piece of evidence that we can use to infer that a decline has actually happened or the population is small. And then sort of to conclude the main takeaways from the project overall, what I found is that hunter-gatherers were impacted quite differently by the spread of agropastoralism, of agriculture and pastoralism in Southwest Ethiopia. So, we showed that some populations experience declines in effective population size, which is sort of the expectation for hunter-gatherer populations. We think of the spread of agriculture as leading to the decline of these populations, so a drop in NE. However, we didn't quite see this in a universal sense. So we see a lot of these Southwest Ethiopian populations with high proportions of hunter-gatherer ancestry actually showing increases or perhaps a steady population size over time. So, overall what we see is that ancient hunter-gatherers probably responded to the encroachment of agricultural and pastoralism in a variety of ways, including responses like cultural adoption. So most of these populations, apart from the Chabu, are no longer practicing hunter-gatherers, as well as through gene flow. And with that, I would like to thank my collaborators, especially Ricky, Justin, and Zach for collecting all the samples, Barry Hewlett and Brenna Hen for advising me, and of course the Chabu, Majang, Shikacho, Bench, and Cheko communities for participating in this research. Final plug, I will be starting my own lab at Clemson University later this year. Also tigers, by the way. So if this is something that you're interested in, please get in touch. I will be hiring graduate students and postdocs in the very near future. And with that, I'll thank you, and I'm happy to take any questions.
Some questions back here.
Hi, thank you for the wonderful talk. I was curious about, so your simulation results were really interesting, and I was curious a bit about, it seemed like the result you have that you showed the slide a few times where the migration happens and it sort of does the opposite of what you might expect to any on the left here.
Yeah.
Right, like might you expect any to pop up when the migration's occurring? And
it seems to be like, I mean, in this case it's before
Right.
I don't know, like it's not really optimized for gene flow to happen, so like weird things are happening, but that's something that we saw. It was either like right at the point where gene flow happened or like a little bit before.
Right.
And I don't really know why that was the case, but
yeah.
Zane has seen that with like PSMC stuff too.
Oh, really?
It sometimes looks like offset.
Right.
It really just handles it. Yeah.
Yeah, anyhow.
Know why,
think,
or just
I'm not entirely sure, but I think it just makes it less reliable in those cases that you can at least point to that point and say, oh, there was something that changed here.
Yeah,
it's like
right.
it actually happened sooner.
Yeah, unfortunately that seems to be.
Looks like we have Uma.
Hi, thanks. Interesting talk. I was wondering, you said that you simulated scenarios with 25% gene flow. That seems really high.
Yeah.
So I was just wondering whether do you have any indication that, when you have colonization or gene flow with agriculturalists, it's kind of a swamping out, and that's why you have these high levels? Or do you have any archaeological ways to think of what bounds there might be on that gene flow?
Yeah, so there are methods, again, based on F statistics for inferring proportions of gene flow from various sources. We found it hard. We had a hard time actually applying those methods, I think mostly because we lacked ideal reference populations. So a lot of those methods, we ran them. They didn't really give us any conclusive evidence. I sort of chose these like 10, 25, and 50% based on sort of bounding the proportions that we see in the admixture plots more than anything else. That was kind of, the rationale there.
Thank you for your talk. I had a question about the admixture plots, actually, and I'm not too familiar with them, so preface
I meant to explain those a little better, sorry.
Oh, no, you're totally fine. I was just curious on the Bayera, individual. It seemed like with them, they had like these kind of blunt lines versus, so I was just curious about that.
Oh, yeah, sorry. So that I just plotted wider because it's only one individual. So here, each vertical bar is an individual. And so I think I plotted it like 10 or 20 times wider just so you can see it. That's the only reason.
Thank you for the very interesting talk. I was wondering about the social dynamics of the Chabu people, and did you happen to look into sex bias dispersal, which might indicate like increased patri or matrilocality with agriculture?
That's a really, yeah. So a lot of the work that we're doing in this region is sort of ongoing, and that's sort of something that we're really interested in looking at in more detail, as well as sort of cultural transmission and how that might be sex bias as well in this population. In a situation where marriage outside of the Chabu ethnicity is becoming more common, we're interested to see if and how, culture and language are actually going to be transmitted. So we do have some data to look at that, but yeah, that's sort of a whole other angle, which is like really interesting for sure.
Thank you.
I've got a quick question. On one of the last slides that you showed, it had the Ari blacksmiths and the Ari cultivators plotted on the same thing.
Yeah.
And it looked like for a little while back in the past, they were overlapping, and then you can see that they kind of diverge. Does that timing match up with things that you know about them? Like, does it seem like it's estimating the timing accurately of these demographic events?
Yeah, so it's interesting, like, to interpret them diverging as also like population divergence, which could be, it could be that they're just sort of similar, but evolving in parallel as well. So the one paper that I saw where they actually estimated the divergence time, it was in the last 4,500 years. So it's not more specific than that. And it's exactly corresponding to the age of the, because they're actually really closely related to, they live like right in that same area as Bayero was found. So it kind of matches up in that sense. It's like, it's about right. It's obviously after 4,500 years ago, but as to something more specific than that, we couldn't really, yeah.
With the IBD any method, you had the sample sizes of 20 and 50, but do you use the larger human data set as a reference population, or are you just using those within a population set to do the method? If that makes sense.
So
I'm wondering if do you need a reference data set for the method of IBD any, or is it you're only using the population itself?
Right. So you only use the samples that are of this population, basically. Okay. So, I mean, of course, like for other steps in applying the method, like phasing and phasing. IBD inference, other references are helpful for that. So yes, that is certainly, although we didn't have a lot of like really good references for this particular part of Africa. So, we use sort of a larger data set to help with phasing and so forth. But for the method, actually applying the method, we infer IBD within the population, and that's all we use here.
Okay, thanks.
I'm just interested from sort of the anthropology perspective. This may not be the part you're involved in, but the anthropologists who work with these folks, are they sort of bringing results back and results sharing, and does it match with their own histories or sense of themselves? Because I imagine it might be kind of alarming to see a plot like that about oneself.
For sure, yeah. So the collaborators that we work with are, they've been working there for a very long time. They go back frequently, which is why we work with them. So we did do a return of results to the Chabu in between, like before it was published. They did not seem to be particularly surprised by this. I think that they are aware that they are being displaced from their lands. There was no real disagreement about them being sort of like the original inhabitants of the forest. Even their neighbors agree with this and understand this. So, yeah, not really.
It sort of matches with their own sense of what's happening.
Exactly. And I'll say that they're more concerned about, like getting access to healthcare and education for their kids more than,
makes sense.
What we're telling them here.
I think there was one. Is there still time for questions?
Let's do one more.
You have to flip it on. Yeah.
Okay, so thank you very much for your talk. I was curious as a follow-up with the reference population. So since it's becoming more and more common and affordable to make region-specific human reference genomes, do you have in your future plan to make one super specific for the Chabu population? Or if not, if there were to be like a Africa-wide one, would it be close enough to infer some valuable information, or is it still like too diverse to gain any more information from that?
Yeah, it's a really great question. So we actually do have, since I sort of finished working on this particular project, more whole genome sequencing has been coming online, and so there's a consortium that I'm a part of that's sort of basically, and there are several now, it's getting a little bit better where they're trying to sample more diversity within Africa. So I do expect that we'll be able to do more in the future with sort of whole genome data sets and just, some more of the like kind of cutting edge methods that rely on that kind of information in the future. But yeah, so for all this data, this was all like done on a SNP chip. So I didn't mention that either, but sort of a commercial human chip. But yeah, there's obviously a lot more we could do if we had whole genome data, and that's forthcoming.
Let's save other questions for the coffee break and give another round of applause. So we're going to take a half-hour coffee break now and then come back for the